There is an alternative to tissue sectioning for studying loose connective tissue. This involves peeling back a piece of skin to get at the hypodermis in a living but anaesthetised animal. The tissue is made slightly swollen by injecting a little saline solution to spread the fibres apart. Then, small bits of the tissue are clipped out and teased apart on a glass slide to produce whole mount spreads instead of sections. The mounts are immediately fixed and stained, and you are viewing such a preparation in this image. What one ends up with is considerably a matter of chance. It's obviously impossible to standardise this procedure, so each spread will be different from every other. I'll begin first by identifying fibre types. The fibres are not cut, but run for long distances until they get lost in a tangle of other fibres or tissues. Elastic fibres behave much like rubber bands. They'll be stretched into straight, thin lines or recoiled into spirals where they've been snapped. They can truly branch and are sufficiently refractile to have an appearance distinctive from the collagen, even with little or no stain. Bundles of collagen fibres look like pale pink ribbons of various widths. You can see the lightly stained nuclei of fibroblasts in large quantities, although their cytoplasm will be so pale as to be generally invisible. These nuclei are spindle-shaped. They will appear oval and pale if you look down on their flat face, but will be small and dark if you looked at them end-on. Fibroblasts develop locally from fetal connective tissue cells called mesenchymal cells. Fibroblasts represent perhaps 40% of the cell population in connective tissue of this sort. About an equal number of cells are histiocytes, or macrophages, which are phagocytes. They represent a separate, unrelated cell type derived from bone marrow instead of mesenchyme. Resting macrophages look very much like fibroblasts. However, after they've consumed something, the cells often can be identified by their cytoplasmic contents without any special stains. You can see them here, and here, and again here. And I'll show you some more in the next two images. If they've been consuming fatty material, macrophages will appear vacuolated. The fat will have been extracted during processing, and the pathologist will refer to them as foam cells. One can play a trick with experimental animals to identify macrophages easily in any part of the body. The trick is to inject a colloid pigment, such as trypan blue, carmine, or India ink, into the peritoneum of a living animal. 1 week after the injection, the animal is sacrificed. Phagocytes all over the body will have phagocytized the injected coloured particles. The rat which generously contributed this tissue for this image received India ink. And the population of histiocytes, or macrophages, in the tissue spread is beautifully marked by the ingested black or dark grey particles. The tissue has also been stained lightly with hematoxylin and eosin to reveal other cells and extracellular components. These long interconnected profiles are blood capillaries, and you can see in some areas the pale stained nuclei of the endothelial cells. Only the histiocytes accumulated the trypan blue to any appreciable extent, so this procedure demonstrates a clear-cut functional difference between the macrophage system and any other population of cells. A third cell type to look for is the mast cell. These cells are large and their cytoplasm is distinguished by large numbers of small granules containing heparin and histamine, among other secretory products. Neutral red was added to the stain for this tissue to dye the mast cells a brilliant red. Mast cells tend to cluster along arterioles and their granules will make the cells stand out. The remaining cell types that you may encounter will be invaders from the bloodstream, particularly lymphocytes and eosinophils, and these will be described in later lectures.